I presented and discussed data on our genomics in myeloma, done here at Dana-Farber with our collaborators from France and England as a joint group, Dr. Avelovazo and Campbell. And to give the summary, I think it is a really great time where we can do all this genomic analysis, and we have done large samples to try to understand various DNA changes, RNA changes, and some of the new modern changes. So part of what I showed was the summary of our DNA data. And to put it in a very simple term that is usable for our clinical purpose, we find lots of mutations. However, no single mutation is more frequent than 20%. Now, these mutations give us new targets. So we have mutations in RAS pathway, RAF pathway, P53. We have inhibitors of RAS and inhibitor of RAF that we can use in clinic. Second thing, we know that there are multiple clones in myeloma, like many other cancer, and these clones change over time. So practically speaking, we do genomic analysis, usually a diagnosis that may not be adequate, because when patients relapse, the disease changes, so we need to repeat it. So unlike what we used to doing, say, last year, the new important change is that we need to repeat genomics periodically to know what is the myeloma at that particular time, not what it was last year or 2 years ago. Third thing, some of these changes are driven by certain processes in the cell, and we can co-opt those processes to be able to kill myeloma cells more effectively. So we can use this genome instability to our advantage for therapeutic purpose. And finally, what I showed is that we are beginning to develop these targeted platforms for genomics, where we don't have to do a huge, big, fancy genomic analysis. This smaller size method can identify all the common mutations. It can look for all the common changes that we can use to prognosticate, find target, and treat the patient in a more precise way. So those are going to become common things for our clinical application in a very near future.